Good day everyone, my name is Dr. Lusia Barek from the Preparatory Center for Science and Technology of Universiti Malaysia, Sabah. Today I am honored to present our research entitled Induction of Cell Death and Apoptosis in MCF-7 Human Breast Cancer Cell by Tongkat or Eurycoma Longifolia Jack leaf extract. This study was conducted in collaboration with colleagues from UMS and UTHM Malaysia. It will be presented as part of the 7th Kuala Lumpur International Multidisciplinary Academic Conference 2025. Let me begin by giving you an overview of the context and motivation behind this study. Breast cancer remains one of the most commonly diagnosed cancers and a leading cause of cancer-related mortality in women across the world. Among various breast cancer types, the MCF-7 cell line is widely used in research. It represents estrogen receptor-positive breast cancer, which is known to respond to hormone-based therapies. However, despite the availability of conventional treatments such as chemotherapy and radiation, issues like drug resistance and severe side effects are persistent challenge. As a result, researchers have been exploring plant-based therapies that are believed to offer fewer side effects and additional therapeutic benefits. The goal is to find compounds that are effective, safer, and potentially complementary to the existing treatments. One plant that has drawn attention is Eurycoma longifolia, which is commonly known as Tongkat in Malaysia and also as pasak bumi in Indonesia. Traditionally, it is known for its medicinal properties and is widely used to improve energy, especially for male, which used to improve the reproductive health and also the general wellness. Most previous studies have focused on its root extract, which has shown anti-cancer and testosterone-enhancing effect. However, harvesting the root raises sustainability concern. Our research shifts the focus to the leaf extract, which is less studied but more sustainable. We prepare the leaf extract in the form of herbal infusion or tea and investigated how different processing methods like fermentation and drying could affect the chemical activity and biological effects, particularly on breast cancer cells. The main objective of our study was to evaluate the cytotoxic and anti-cancer effect of Eurycoma longifolia leaf extract, specifically in the herbal tea form on MCF-7 human breast cancer cell. We also wanted to understand how different processing techniques such as fermentation and drying methods, namely freeze drying and microwave drying, influence the bioactivity of the extract. In particular, we focus on comparing the cytotoxicity of different extract types, identifying the apoptotic mechanism triggered by the most potent extract. This approach not only aimed to validate the potential use of Tongkat as anti-cancer agent, but also emphasize the practicality of using leaves in a sustainable manner. We began by preparing 4 types of leaf extract. The first one, unfermented freeze-dry, second, unfermented microwave dry, third, fermented freeze-dry, and fourth, fermented microwave dry. There were also 2 other commercial tea of true tea Camellia sinensis as the comparison to the Eurycoma longifolia sample. Then we performed a series of cell-based assays using MCF-7 cells. The MTT assay was used to measure cytotoxicity, or how toxic each extract was to the cancer cells. From MTT assay findings, the most potent Eurycoma longifolia extract was selected for further analysis to determine the possible anti-cancer mechanism of action. In this analysis, at first we used the acne fragmentation analysis to see whether the cells were undergoing programmed cell death. Then, cell cycle distribution analysis helped determine whether the extract caused cell cycle arrest or not. The apoptosis assay using annexin V were used to quantify early and late apoptosis. To explore molecular mechanism, we measure the activity of caspase-8 and cytochrome C, as well as the expression of key apoptotic related genes like p53, BEX, and BCL2. Moving on to the result and discussion, we find that using our true MTT assay, the result shows that the unfermented freeze-dried extract exhibit the highest cytotoxicity against MCF-7 with the lowest IC50 value. This means that lower concentration of this extract was required to kill 50% of the cancer cell, making it the most potent among all the tested samples. The reason behind this may lie in the processing method. For the unfermented leaf, steam blanching was used to deactivate polyphenol oxidase, an enzyme that breaks down phenolic compounds. This step helps preserve phenolic content, which is important for maintaining cytotoxic activity. In contrast, the fermented leaves were not steam blanched, allowing more phenolic degradation during fermentation and drying. Additionally, withering and oxidative fermentation may further reduce these beneficial compounds. Freeze drying is a non-thermal method that preserves delicate phytochemicals better than microwave drying. It retains cell structure and bioactive compounds such as phenolic acid and flavonoids by forming the crystallized leaf cells, which often retain intracellular compounds in the most stable form and potentially protecting sensitive phytochemical from degradation. In contrast, microwave drying involves heat, which might degrade or change the structure of these compounds, reducing their effectiveness. When we analyze the DNA fragmentation patterns of MCF-7 cell after treatment with the most potent extract, a typical feature of apoptosis is the ladder-like pattern of fragmented DNA, which we observe clearly in our result. This pattern was similar to the positive control group treated with doxorubicin, a known anti-cancer drug. The presence of these bands, ranging from the small to the large fragment, indicates that programmed cell death had occurred in response to the extract. Next, we examine the distribution of cells across the different phases of cell cycle. We found a significant accumulation of cells in the sub-G1 phase, particularly at 24 hours, 48 hours, and 72 hours after treatment. The sub-G1 phase is associated with apoptotic cells that have lost DNA content due to fragmentation. This accumulation shows that the extract disrupts the normal progression of cell cycle and pushes the cell towards apoptosis instead of allowing them to divide or repair themselves. This is an important mechanism because arresting the cell cycle is a key strategy in preventing cancer cell proliferation. To dig deeper into the mechanism, we look at two critical players in apoptosis, caspase-8 and cytochrome C. We found that caspase-8 activity was significantly high or increased at 24 hours, suggesting early involvement of the extrinsic apoptosis pathway, possibly triggered by the external signal. Meanwhile, cytochrome C activity increased over time, with two-fold rise by 72 hours, indicating mitochondria-mediated intrinsic apoptosis was also activated. So these findings suggest that the extract activates both extrinsic and intrinsic apoptosis pathway in the MCF-7 cells. Next, for the expression of apoptotic protein p53, BAX or BEX, and BCL2. From our result, it shows that the BCL2 as an anti-apoptotic protein decreased over time. In contrast, p53 and BEX, which promote apoptosis, increase at 24, 48, and 72 hours. This shows an antagonistic pattern. As BCL goes down, p53 and BEX go up, indicating that extract trigger apoptosis by activating pro-apoptotic signals while suppressing survival signal. One notable characteristic of MCF-7 cells is the absence of functional caspase-3, a key executioner in apoptosis. Despite this, we observe DNA fragmentation and apoptosis, indicating that alternative pathways were activated. Based on our findings, the possible mechanism involved the first one, caspase-8, initiating the extrinsic pathway, leading to apoptosis through caspase-7, or other effectors such as caspase-6. Mitochondrial stress leads to p53 activation, which upregulates BEX and downregulates BCL2, causing the release of cytochrome C. And activation of caspase-9 then activate the caspase-7 as the executioner caspase. This dual pathway activation, even without caspase-3, supports the strength of the extract in inducing controlled cell death in the cancer cell. So both pathways cause DNA fragmentation, a clear sign of apoptosis. Even without active caspases, MCF-7 cells can undergo caspase-independent apoptosis. Protein like AIF or endonuclease G move from the mitochondria to the nucleus and break DNA. So even without caspase-3, apoptosis still occurs, and DNA fragmentation is a key indicator. To conclude, the unfermented freeze-dried leaf extract of Eurycoma longifolia successfully induced apoptosis in MCF-7 breast cancer cell. The processing method, especially freeze-drying, plays a vital role in preserving the extract bioactivity. This finding contributes to the understanding of how herbal tea made from Tongkat ali leaves could be developed as a complementary or alternative anti-cancer therapy. Further in vivo studies are necessary to explore its safety, toxicity, and bioavailability in animal or human models before clinical application can be considered. These are some of my reference for this study. Thank you for your attention. I hope this presentation provided valuable insight into the potential of Eurycoma longifolia as a natural cancer fighting agent. If you have any question or would like to collaborate, please feel free to contact me at luciamoses@ums.edu.my. Thank you once again, and I welcome any feedback and discussion.